Good day, folks. Welcome to my channel. In this video, I will be discussing the second part of mutation. In the previous video, you have seen about the different types of mutation. So, in this video, you will see the effect each type of mutation causes. So, the first one is silent mutation. So, here a point mutation may change the codon for one amino acid to a synonym for the same amino acid. So, that is here there will be change of codons, but the change will not produce any effect. So here you can see that one example is if CUA is mutated to CUC. So both these codons code for the amino acid leucine, and so this mutation has no effect and thus produces no net effect on the phenotype. Thus the mutation is silent. Even if it is taking place, it is not bringing about a change in the phenotype. Okay, so the next one is missense but acceptable mutation. So here a change. So the silent mutation is an example of substitution mutation, and missense is also an example of substitution mutation. So here a change in amino acid may be produced in the protein but with no functional consequences. So let's see an example to understand what it means. So here in the normal HbA molecule, that is a hemoglobin molecule, the 67th amino acid in beta chain is valine. Now the codon in mRNA is GUU. Now if a point mutation changes it to GCU, the amino acid becomes alanine. So this is called hemoglobin Sydney. Now this variant is functionally normal. A conserved mutation occurs when the altered amino acid has the same properties of the original one, example glutamic acid being replaced by aspartic acid. So here also a phenotypic variation is not seen. But the difference you have to understand between silent mutation and missense but acceptable mutation is that here there will be mutation and change in the codon, but the change and the original codon both will be coding for the same amino acid. Now here in the case of missense but acceptable mutation, there will be a change in the codon that is due to mutation, and that new codon will be coding for another or a new amino acid. But this new amino acid have the same functional properties so that the phenotypic difference is not seen. Okay, now the third type is missense but partially acceptable mutation. So here in this type, the amino acid substitution affects the functional properties of the protein. So HbS or sickle cell hemoglobin is produced by a mutation of the beta chain in which the sixth position is changed to valine. So instead of the normal glutamate, so the glutamate or the glutamic acid is converted to valine or changed to valine. So that is what is happening in sickle cell hemoglobin. Now here the normal codon GAG is changed to GUG, and that is transversion. That is here adenine, which is a purine, is converted to uracil, which is a pyrimidine. So it's a transversion. Now this HbS has abnormal electrophoretic mobility and subnormal function at low PO2, so leading to sickle cell anemia. So here in missense, there is difference in the functional properties of the protein. So there is no need to confuse with the first two. Now the next type, the fourth one, is missense but unacceptable mutation. So here the single amino acid substitution alters the properties of the protein to such an extent that it becomes non-functional and the condition is incompatible with normal life. For example, HBM results from histidine to tyrosine substitution of the distal histidine residue of alpha chain. So the other two examples which we have discussed before, it is for the beta chain. So here we can see the alpha chain of the hemoglobin. So it leads to the formation of methemoglobinemia. Now different variants of HBM may result from a similar substitution of distal or proximal histidine residues of alpha or beta chain. So the difference in this type of missense is it is completely unacceptable mutation. That is, here the single amino acid substitution alters the properties of the protein to such an extent that it becomes non-functional, and the resulting condition will be incompatible with the normal life. Okay, now the fifth type of mutation is nonsense or terminator codon mutation. So here a tyrosine, that is having the codon UAC, may be mutated to a termination codon. You know that UAA, UAG and UGA, they are the terminator codons. So if the C is converted to A or G, That is, if the cytosine is converted to adenine, that is mutated to adenine or guanine, so there is a mutation, and these are the termination codon. So if there is a termination, like mutation resulting in the formation of a termination codon, this will lead to the premature termination of the protein. That is, the protein will get terminated whenever it comes in contact with the termination codon. So if there is a mutation of tyrosine to a terminator codon, then there will be premature termination of the protein, and so the functional activity may be destroyed. So example, it is seen in beta thalassemia. Now another case is if a terminator codon is altered into a coding codon. That is, if a terminator codon such as UAA or UAG, so here you can see UA, if it is converted to or mutated to CAA, so this results in elongation of the protein to produce run-on polypeptide. That is, the protein will not be having a termination. So there will be an increased protein structure, so that is called run-on polypeptide. Now the above six the above six mutations, it is produced due to the substitution mutations. Now if there is a deletion or insertion of bases, let's see what happened. So we can see the frame shift mutation. So this is due to addition or deletion of bases. So from that point onward, the radium frame shifts. A garbled, that is completely irrelevant protein with altered amino acid sequence is produced. So here you can see a example. So here in this hypothetical example, deletion of one uracil changes all the triplet codons thereafter. Here you can see the normal protein which is produced from the normal codons. But if there is a cancellation of a uracil, that is, here you can see this U is removed. That is, there is a deletion of a base, then the radium frame is completely changed and a completely irrelevant protein may be produced, and therefore useless protein or no protein is produced. Now this frame shift mutation can lead to thalassemia due to premature chain termination or run-on polypeptide that are non-functional. So either, like, if there is a change, you might get a termination codon earlier or you might not get a termination codon. So it can lead to even premature chain termination or run-on polypeptide. So all these mutations that I have discussed above are the main mutations. Now next you have another type, that is a conditional mutation. So this is the one that you can talk in a general sense. So here most of the spontaneous mutations are conditional. That is, they are manifested only when circumstances are appropriate. Bacteria acquire resistance if treated with antibiotics for a long time. So this is explained by the spontaneous conditional mutation. In the normal circumstances, wild bacilli is a type of bacteria will grow. So in the medium containing antibiotic, the resistant bacilli are selected. Now in a tuberculosis patient, a lung cavity may harbor about 10 raised to 12 bacilli. Now this may contain about 10 raised to 6 mutations, out of which a few could be streptomycin resistant. Therefore, if only one drug is given, there will be overgrowth of drug-resistant bacilli. So to avoid this, two anti-tuberculosis drugs are usually given together. So the main thing in conditional mutation is, with time, in the case of bacteria, there will be mutations if the same antibiotic is applied and the bacteria will get resistant to a particular type of drug. So drugs are given in mixture rather than given individually, because the statistical probability of a single bacillus acquiring resistance against both drugs is negligible. That's it for this video. If you honestly did understand the topic, do like, share and subscribe. Thank you.